Machines. They enable us to be creative and produce spectacular structures. We also find magnificent structures in nature, and each of these living structures was also built by machines. Machines that are far more elaborate than anything that humans have ever built. In fact, this log is currently being decomposed by tiny fungi that are vastly more complex than his drone. I'm Mike Behe. Welcome to the unseen world of organic micro-machines. Every living thing in the world is made of cells. Microbes are single-celled organisms. Advanced life forms have trillions of cells. Inside a cell is a bustling operation of interconnected molecular machinery so sophisticated it would put any modern high-tech factory to shame. This intricate machinery is what gives the cell its remarkable abilities to process energy, execute genetic instructions, and replicate, all with unbelievable efficiency. I'm a biochemist, a scientist who studies the molecules of life. There's never been a more exciting time to be a life scientist. This is a golden age of discovery in molecular biology. Stunning breakthroughs in our understanding of the most fundamental unit of life, the cell, have been coming faster and faster. My own view of the cell took a turn years ago. I was in a lab at the National Institutes of Health doing postdoctoral research. I was discussing the origin of life with a fellow postdoc. As she and I thought about the cell, we wondered, how could its complex membrane, proteins, metabolism, genetic code, how could all that have formed by the accumulation of undirected changes? So we were both sort of stunned by the notion, but then we just laughed it off. We figured that even if we didn't know the answer, somebody must know. Well, in the 10 years after that, I didn't come across any science publications that adequately answered the nagging question, exactly how did the cell get to be so complex? Everything I read presumed that those biological mysteries would eventually be solved by Darwin's theory of evolution. Being an evolutionist myself, that was fine with me. Then I came across a book titled Evolution, a Theory in Crisis by a geneticist, Michael Denton. It addressed profound problems with Darwin's theory, and it showed that science didn't have the answers. No matter that everyone assumed it did, or assumed that it would eventually, I began to look at biology with a fresh set of eyes. And I started asking questions, not about the why or when, but about the how of biological origins. We all love cars for their beauty and performance. But except for guys like these, not everyone really understands what's inside. In a way, cell biologists are like auto mechanics. Each cell has an astonishing number of intricate parts, way more than any car. We study what the parts are, how the parts work together, and even which parts of the cell can be left out without causing too much harm. If you understand the inner workings of the cell, you can understand a lot about all living things. What makes them thrive, what makes them fail, and what causes them to evolve. You and I are made of about 40 trillion cells. They comprise every part of our complex human anatomy. But the truth is that we can reduce the number of parts and still have function. If we lost an eye, an ear, or a hand, it would be very unfortunate, but our lives could go on. The same applies to this rather complex drill. It has some cool add-ons, but I could take off this battery gauge, or this LED light, or a few other gadgets, and I'd still have a working drill. But how far can I reduce its complexity and not ruin it? I don't want to take apart my drill, so let me move to another example, this mousetrap. The complexity of this machine entails 5 parts: the platform, the spring, the hammer, the holding bar, and the catch. Now, if I reduce the number of parts from 5 to 4, if I take away the holding bar or the spring or any other part, it wouldn't work. Every part is essential. I call this irreducible complexity. So does this apply to biology too? Do we have examples of irreducible complexity in life? Try this one. Here's a bacterium, a single-cell microbe. Some bacteria have a flagellum, which is like an outboard motor on a boat. It rotates to propel bacteria through liquid, and it's necessary for their survival in order to swim to search for food. A flagellum has a number of parts: a drive shaft, a universal joint, a rotor, bushings, stator, even a clutch and braking system. The motor of the flagellum has been clocked at 100,000 revolutions per minute. And as with the mousetrap, removing even one component of this elegant machine destroys its function and renders the bacterium, shall we say, dead in the water. The flagellum is irreducibly complex. Now we know that this was assembled in a factory, but how is the bacterial flagellum developed in nature? Keep in mind, the flagellum is irreducibly complex. All the parts have to be in place, or it does not work. Could it have been developed blindly, in stages? Let's say in the distant past, the bacteria first gained a drive shaft. Later on, a rotor, then a stator, and then the rest. Then the parts got in sync as a high-performance engine. The theory of evolution tells us that given enough time, any organic structure we see today, no matter how complex, could have slowly and fully developed by random, unguided forces. We don't typically ponder the elegance of bugs, especially the annoying ones. But scientists and engineers have come to realize they are actually mechanical marvels. Nothing we humans have ever built even comes close. In fact, a growing field of engineering called biomimetics is making breakthroughs by mimicking biology. For example, studying the eyes of flies led to the development of advanced optical sensors. The physics of dragonfly wings allowed increased efficiency of wind turbines. Cockroach mechanics paved the way for increased agility in robots. A honeybee algorithm helped to optimize the internet. And the tiny plant hopper is helping scientists to maximize the strength and rotation of gears. I thought humans invented gears. Those perfectly precise, interconnected drivers that have transformed machinery for centuries. Well, it turns out that biology is far ahead of our engineering. The plant hopper launches like a rocket and can jump more than 100 times its body length. That's like an Olympic long jumper hurtling across 2 football fields. Using special cameras, scientists at Cambridge University discovered that the plant hopper coordinates the motion of its legs with these incredibly precise gears. Making long leaps accurately requires that the legs be in perfect sync. If one leg flexed even a fraction of a millisecond before the other, the insect would lose power and tumble erratically. As one leg begins to move, the interacting gears start the other leg too. With the teeth engaged, the gears spin at a whopping 50,000 teeth per second, and the bug is propelled with maximum power and coordination. As science learns more about the structures of living organisms, we seem to be finding limitless examples of mechanisms that are both precise and purposeful, both remarkably configured and beautifully functional. So what accounts for the stark purposiveness of the machinery of life? For over 150 years, evolutionary biologists have thought they had an explanation: Darwin's theory of evolution. Darwin proposed that all life evolved simply by variation that occurred within a species. Then, natural selection determined which ones survived. For example, let's look at bear evolution. Scientists believe the polar bear in the Arctic evolved from ancestral brown bears in adjacent regions. Here's a possible scenario. Many thousands of years ago, a bear cub was born with a mutation in its DNA. The change gave the lucky bear the ability to eat a high-fat diet, like seal blubber. So the mutant bear and its descendants could hunt seals for food. Generations later, a baby bear was born with another mutation that altered its color. The subsequent change from brown fur to white was useful in the snowy environment for sneaking up on prey. More such helpful mutations followed. So variation in the ancestral species of the brown bear, followed by natural selection for the harsh, snowy surroundings, were responsible for building the magnificent polar bear in small steps. We can see fascinating evolutionary changes in species, but modern science now knows something that Darwin didn't know, that the changes in a species that we can see are driven by molecular changes in genes and DNA that we can't see, at least not with our eyes. What precisely are those helpful mutations in DNA? How exactly do genes change? If brown bears can give rise to polar bears, does that mean we'll see future genetic changes making new species? Will we humans mutate into superpowered X-Men? Dogs are easily one of the most diverse species on Earth. Geneticists tell us it's likely that each of them descended from one animal, the wolf. These breeds look very different from each other, but they all share much of their DNA with those early wolves, and that's what makes them dogs. Now let's move inside the cell and see how this works. DNA is found in the nucleus of the cell. It consists of a long chain of chemicals designated by the letters A, G, C, and T. Some sections of DNA are called genes. They contain the specific instructions for making the proteins that form the cells that give the dog its traits. In our last episode, we saw that DNA in brown bears had mutated to form polar bears. Well, how does that work? A mutation in a gene occurs when there's a change in its sequence. Often this results in breaking one of the instructions for forming the animal. In this case, the mutation was in the specific gene that determines fur color. The disabled gene is unable to give instructions to make colored fur, so the mutant fur is white. The change in bears occurred long ago. Wouldn't it be great if we could observe mutation today, in real time? Well, we're in luck. Critical work has been done in the laboratory of Richard Lenski, who worked with a species of bacteria called E. coli. The Lenski lab has been growing generations of E. coli in flasks for 30 years. In the early 1990s, they saw that the descendant bacteria began to grow faster than their ancestors. That was great news for the happy E. coli, but why was this happening? A decade later, they were able to determine the mutation that caused it. What they found was that a certain gene in the E. coli had been destroyed. Losing that gene ended up helping the bugs grow faster. Then they looked at a dozen other helpful mutant genes in the E. coli and saw that they too had been broken. Mutations had left the genes either crippled or completely disabled. But that seems odd. How can breaking a gene help the organism? Imagine you have to drive to a destination on one tank of gas, or you'll die. Given the weight of your car, you'll never reach your destination on just one tank. So you toss out the hood, a seat, floor mats. the lighter, degraded car gets you to your destination and you survive. So breaking genes can help E. coli adapt to a lab environment, and broken genes help the polar bear adapt to the Arctic. How about our dog friends? In just the past 20 years, scientists have actually discovered many mutations responsible for traits of breeds. And again, the mutations don't construct new genes. Most of them break or damage pre-existing genes. For example, increased muscle mass in some breeds derives from degradation of a myostatin gene. We also know what mutations caused a yellow coat, short tails, even the lovable friendliness of dogs towards humans compared to less friendly wolves. So a dog can be changed by breaking genes, but it comes at a high cost. The broken genes very likely will never return to their original working state. The dachshund with short-leg genes won't be returning to anything resembling a Labrador. And we won't see the polar bear mutate back to its old self eating berries. In short, helpful mutations are not a DNA upgrade. Getting a newer smartphone is an upgrade. It's more helpful because it has completely new features. But mutations don't install new features in DNA. They only make changes to existing ones. A mutation is more like disabling your GPS. It may help to save your battery, but it doesn't add a new function. We're told that random mutation is the main driver for evolutionary change, and that evolution is responsible for lower forms being upgraded to higher ones. Yet the latest scientific results show new species are made by breaking genes, by devolution, not evolution. As we search for the X factor that accounts for the complexity of life, we have to follow the evidence. Let's begin with the cell. What was a mystery to Charles Darwin is now well understood. We know that the cell encodes and transmits information that regulates the size and shape of living things. And we know the cell is comprised of insanely complex machinery. What's more, much of it is irreducibly complex. All the parts are necessary in order for an irreducibly complex system to work. Irreducibility is evidence that all the parts were formed intentionally, with the end purpose in mind, not randomly over time. Finally, Darwin's explanation for complexity was evolution, where random small variations over many generations progressively led to life of greater and greater sophistication. But science has just recently discovered that variation comes from genetic mutation, and that even helpful mutations usually break genes. That's not evolution, that's devolution. So what is the evidence for some X factor that accounts for biological complexity? If you were to come across this formation for the first time, what could you conclude from examining it? There's no apparent evidence that shows when it was made, who made it, or how it was made. But we do have clear evidence that it was not arranged randomly. It was planned by an intelligent being. The purposeful arrangement of parts, that is the way, the only way that we recognize the work of a mind. And the more intricately the parts are arranged, the stronger the evidence that a mind was at work. The X factor that accounts for complexity is intelligence. An intelligent being is responsible for much of life. The exquisite and purposeful design found throughout nature is overpowering evidence. You might say, hold on a second. Don't we have to see the arrangement being put together to decide for sure that it was done on purpose? After all, we can see how an intelligent builder constructs a home, If we can't see or understand how design occurs in nature, why should we believe there is an intelligence behind it? Well, it's what we can see that is the whole point. Suppose 3 visitors from a distant place stumbled across this surprising stone configuration that they had never heard of before. Not knowing how it was formed, one visitor might think it had been chiseled. The second guesses that power tools were used. The third might suppose it was cut by lasers. But none of them would conclude that Mount Rushmore was formed accidentally through gradual random erosion by wind and rain. They would immediately know it was purposely designed just by perceiving the intention behind the work. The purposeful design of nature was nearly universally held common knowledge for millennia. Some thinkers, such as the philosopher Aristotle, believed that purpose was built right into nature. Others, such as Galen, the esteemed physician in Rome, thought that purpose had been added to nature. But almost everyone, educated or not, religious or not, realized that life was intentionally designed. Going beyond biology, other fields have acknowledged purposeful design through the ages. Astronomy, chemistry, and physics all reveal arrangement and order. Science took a detour from that strong consensus when Charles Darwin proposed random, unguided evolutionary causes. Now we know that Darwin was right for the case of small changes in life. But the void of evidence for large, random evolutionary changes supports the enduring consensus of purposeful design. So, who is this mind? One thing we know is that it must be phenomenally intelligent to be able to design life. What's more, a mind is at least as intelligent as its work has shown. It's likely much more intelligent. But it's clear that errors do occur in nature. Babies are born with birth defects. Good people get cancer. Why? As I said earlier, I count myself among those biologists who think that science alone may not be able to answer the question of who or why. Perhaps philosophy and theology are needed for that. But the identity of the designer is a separate question. The bottom line is that just like those travelers encountering Mount Rushmore for the first time, we can see the evidence. If we carefully study it, we can know with certainty that life was designed by a mind. The discussion continues on the series website. Please go there for a deeper look at these topics. This man has just suffered a serious stroke. His best chance for a full recovery is to receive proper medical treatment within 90 minutes. A 911 call alerts a dispatcher. The dispatcher alerts the local emergency medical technicians. The EMT determines it is likely a stroke. The call is made to the hospital. The doctor notifies the team that the patient is inbound. When the patient arrives, he receives rapid, stroke-specific care. Today we're going to look at a similar emergency response system, one that has assisted in my own life, and in your life too, many times over. A cut in a person's skin allows blood to flow freely, but with a cut this size, in a short time, the bleeding stops on its own. How does it do that? The answer is not that the blood just dries up and plugs the hole. It stopped because of a sophisticated emergency response system operating under her skin. It's called the blood clotting cascade, and it's one of the true wonders of human physiology. Here's an overview of how it works. Here's the cut from the thorn. As the red blood cells flow out, there's nothing to stop them. This does not look good for our gardener. But watch this. These are chemical signals. It's a 911 call. And here come our body's first responders. They're called platelets. They might not look impressive, but they're actually mighty morphing heroes. As they land, these remarkable transformers stretch out and begin to form a plug. They also release more chemical messengers to summon reinforcements. More platelets arrive, and eventually the outflow is slowed. Whew, it's a good start, but this barrier isn't strong enough to hold for long. We actually need a different team of transformers to save the day. The system activates another series of chemical signals. To the rescue comes an absolute marvel of molecular biology, fibrinogen. These guys do serious work, and their powers need to be used judiciously. So, at the correct time, thrombin shows up to activate it. The fibrinogen molecules then transform into fibrin. Now, watch as the fibrin link end to end to make strands, and they form a super secure mesh. This process is called coagulation. Thanks to the quick work of our elegant response team, the gardener won't bleed out, and the disaster is averted. That is, unless coagulation continues and the expanding clot blocks her blood vessel. Not to worry, other heroes join the fray. Two of the strongest are antithrombin and thrombomodulin. They switch off thrombin, and the fibrinogen is not activated to become fibrin. Later, the cleanup crew arrives. A critical component is plasminogen. When activated, it becomes plasmin. Plasmin is an eating machine. It dissolves fibrin once its work is done. Finally, her body heals the wound and the clot dissolves away. So think about that next time you cut yourself shaving. It's really a beautiful, finely tuned system. Now, the blood clotting cascade has been the subject of rigorous scientific research for many decades. How might the amazing cascade have evolved? In 1970, the plight of Apollo 13 riveted the world.
What it was. Okay. Houston, we've had a problem.
A small defect in an oxygen tank led to a system failure, and the mission almost ended in complete disaster. We've been admiring the blood clotting cascade, but just as with a spacecraft, defects can occur in the system, and the results can be life-threatening. We know that the success of the cascade depends on each component doing its job. Earlier we saw fibrinogen, but there are actually 11 more proteins that play an essential role in the process. In all, there are 12 proteins that contribute to clotting. We also have the anti-clotting proteins. We saw fibrin. There are 10 more on this side. It's a delicate balance, and a defect on either side can result in a system failure. Here's two examples. Some people are born with hemophilia. This disease is caused by a mutation in factor VIII. It leads to insufficient blood clotting and can result in dangerously excessive bleeding. Factor V Leiden thrombophilia is another genetic disease. It's caused by a mutation in factor V. In this case, the protein isn't switched off at the proper times. The result is abnormal and harmful blood clots. Now, let's look at how these defects occur. DNA is the control center of the cell. It contains the genetic information that determines the size and shape of proteins that assemble together to form you and me. A mutation is a change in DNA, which leads to a change in the structure of a protein. Here are three possible effects of mutation. Harmful mutations lead to mild or severe health problems. Neutral mutations basically have no effect on health, and a person might not even know they have one. How about helpful mutations? Are there beneficial mutations that drive evolution? Darwin's theory of evolution claims that life forms advance through small, random, helpful changes over time. Neutral mutations don't cause significant changes. Harmful mutations are damaging, not constructive. For life to evolve, we would need beneficial mutations. Sticking with today's subject, can we find any beneficial mutations occurring in blood? Well, that's easy. I can tell you in no uncertain terms that the answer is yes and no. Adults and children in many regions of the world suffer from malaria. A fraction of people in these regions have a condition called sickle cell trait, which is caused by a mutation that leads their bodies to produce slightly altered hemoglobin. In turn, that can cause their red blood cells to take the shape of a sickle. It also makes them resistant to the malaria parasites, which gives them a degree of natural immunity to malaria. The sickle cell mutation is often pointed to as a clear example of evolution, and so it is. After a fashion, a mutation made a change in the blood cells that helped people adapt to an environment with a terrible endemic disease. Those without the mutation remain vulnerable to malaria. Those points are true, but there's more to the story. In its strongest form, sickle cell also results in anemia, severe swelling, and infections. While it does help with malaria, the bottom line is that sickle cell itself can be very harmful. Sickle cell is just one example of mutations that are ultimately harmful. In fact, the overwhelming majority of non-neutral genetic mutations are damaging, not helpful. So, no, there is no evidence that beneficial mutation results in constructive changes for advancing evolution. What is a system? A system is individual components that are interconnected to perform a task. As we see here, a well-constructed sequence reaches the target. But a critical path that has a flaw will fail. We saw earlier that the blood clotting cascade is a highly complex system indeed. The many proteins are interconnected and interdependent. Each one makes an essential contribution to the system's success. So could such a complex, carefully calibrated system have developed through Darwinian evolution? According to Darwin, those small incremental steps that supposedly advanced life over time occurred randomly. Here's an example of that approach.
All right, next one, looking for number B5. B5, anybody got B5?
Suppose we change the rules of our game. Instead of adding pieces intentionally, our players have to build their systems randomly. First one to the target wins.
N24. Come on, folks. Who's got a system?
Before long,
G4.
our players are ready to find a new game.
I-19.
While relying on chance can be fun for some games, it's no way to build a functioning system. That shouldn't be hard to see.
Bingo?
In the 1990s, I wrote a book titled Darwin's Black Box. In the book, I argued that the blood clotting cascade could not have developed through unguided evolution because the system is irreducibly complex. In other words, all the parts are necessary. Even one missing component will result in a system failure. Instead, the blood clotting cascade had to have been purposely designed to be functional. In the book, I also noted that no science publication had ever shown how the blood clotting system could have been produced through random mutation and natural selection. Little did I know that what I wrote would lead to one of the biggest jolts in my career in biochemistry. A distinguished scientist and leading expert on the subject of blood clotting, named Russell Doolittle, challenged my position. He pointed to a paper about clever experiments with mice, where parts of their blood clotting cascade were removed, and the mice had no ill effects. From that, Doolittle concluded that not all the parts are needed, so the system is not irreducibly complex. His published report was a huge blow to my claims, especially coming from a scientist of Doolittle's stature. So it appeared that the course of my argument could be headed for a major correction. Mouse blood can clot without the full cascade system intact. Or can it? There's no doubt the findings in the paper were the result of valuable, well-executed research. But it turned out Professor Doolittle had misread the paper, which, to be honest, isn't hard to do. Technical papers can be unclear and difficult to decipher. Although removing parts from the mice's clotting cascade did have some interesting results, in the end it made them quite sick, with many of them hemorrhaging and dying. The lesson from this incident is not that scientific papers can be hard to read. Rather, the overwhelmingly important lesson is that not even the best scientists in the world know how unguided changes over time could have built complex biological systems. Of course, blood clotting is just one of many elaborate systems, all of which rely on interdependent components. And it's irrefutable that even the most advanced of all our human-engineered systems comes nowhere close to the intricacy of what is operating right now within you. The complex biological systems within each of us were not assembled by happenstance. They work with dazzling precision because each one has been purposefully designed. What's stored inside here is the most valuable product on planet Earth. And there's so much inside, you could never count all of it. What's intriguing is that the product has no color, no odor. Actually, it has no mass. You might have guessed it. This is a data center. A massive repository of information. It's home to documents, videos, even military secrets. And this is just one of thousands of data centers located across the globe. As you can imagine, removing these sites could have a devastating effect on society as we know it. The same is true in biology. Without the information found in DNA, all life would cease to exist. The unlocking of DNA has revealed information and discoveries that have forever changed our world. We can explore our personal heritage, identify medical predispositions, and even solve crimes. But DNA is much more than a data resource. DNA is the main data source for the physical features of all life. What exactly is information? Why are incalculable amounts of it necessary for life? And lastly, where does it come from? First, what is information? You can think of the fundamental unit of information as one single simple decision. Shall I flip the light switch on or not? More complex decisions require more information processing. Colossal tasks require thousands of decisions, and enormous information management through spreadsheets, diagrams, and instruction manuals. Life also contains written instructions in DNA to build its structures. Let's briefly review how it works. Information in DNA is stored as a sequence of 4 chemicals. They're arranged like letters in a book, billions of them, that can be read and interpreted. Within DNA are segments called genes. A gene is the coded instructions to make a single kind of protein. A machine called RNA polymerase copies a selected portion of DNA into messenger RNA. This is the key. the digital blueprint for building a protein. The mRNA then interacts with another machine called a ribosome. It reads the code in mRNAs and uses it to produce the specified proteins, the building blocks of life. Ribosomes are universal machines because when they're given the right mRNA instructions, they can build any protein. In some ways, it's like 3D printing. Using a lot of information, good programmers can design elaborate parts. Your DNA has the information for manufacturing more than 20,000 different proteins that can be arranged with other molecules to make about 200 different types of human cells. The cells are custom made to form the tissues that form each of your organs and systems. All this uses only a portion of the information in DNA. Fortunately, your DNA can store enormous amounts of data. In fact, it's the envy of every information technologist. Nothing humans have ever designed comes anywhere close to the storage capacity of DNA. Why is so much information needed to sustain life? The first part of the answer lies in the interior of the cell, where enormous amounts of information are constantly being exchanged. Those precisely manufactured proteins fit perfectly to form complex structures. How complex? Some proteins form machines, highly sophisticated machines. Like this one, ATP synthase. These elaborate rotary motors are harvesting energy to power your cells. Or the kinesin motor protein. Yes, it's walking as it transports cargo across the cell. These and many more exquisite machines are operating right now in cells in every part of your body. Now let's move to the exterior, where we'll see that each cell type is part of a massive collaboration of trillions of cells. Individual cells communicate with the cells next to them, passing vital information back and forth. But information also flows between cells in entirely different parts of the body, through chemical and electrical signals. These data networks are what inform your many interconnected organs and systems. What's more, in much of life, as they say, timing is everything. A whole lot depends on the right information in your DNA being activated at the right time. For example, when you were a few months old, your DNA was activated with instructions for growing teeth. Years later, instructions were switched on for a second set of teeth. Then, when the full set came in, the code for teeth was switched off. The genetic program for elephants is different. They get the go code for 6 to 7 sets of teeth before it's finally switched off. So how are these on-off switches flipped in DNA? Here's the most common way. This is one of those chemical signals we showed earlier. Its job is to connect with a control protein and switch it on. The protein can now switch on a control element which resides near genes or in spaces between them. Given the go code, RNA polymerase is enabled. The genetic instructions are copied, and mRNA delivers them to make the needed proteins. Your cells are constantly receiving messages to switch specific genes on and off. And it's remarkable how cells adjust to different situations. For example, when resources are scarce, sometimes an energy-saving mode can be switched on. Warning of predators prompts the defense mode. Cells can even play the noble role of self-sacrifice to benefit the common good. And, in the case of stem cells, when asked, they can transform into a different type of cell. Given all the processing we see in the cell, it's easy to understand why gigantic loads of information are required for life. But how much information are we talking about? The 4 chemicals in DNA come in pairs, A with T and G with C. The sequence spells out the information in DNA. The full sequence contains about 3 billion base pairs. In book form, that would be about 1 million pages. DNA is sometimes referred to as the instruction manual for life. That would be a pretty hefty manual. But as a biologist, it's fun to imagine such a book. I have a manual for my car. What if we could assemble a manual for biological parts? In no case does science yet have a complete description of all the information needed to build even one cell, let alone more complex biological objects. But using what we know about information, imagine the directions for building an organ. A manual for building, let's say, the human eye. Perhaps one of the most complicated things that exists. That one's probably a little too ambitious. How about the heart? It has way fewer parts. Electronics, hydraulics. How about bones? That looks doable. Here's a femur, your thigh bone. Let's give it a try. Bone growth begins in the womb. Step 1. We have designated cells that are instructed to form not bone, but soft cartilage. Next, minerals are added to harden portions, but not all of the bone. Now we add superstructures. These are critical because muscles and tendons need secure points of attachment. Then we move on to a newborn. At this stage of life, the instructions become tricky. Your femur must remain strong, but it also has to grow much longer. The ends of the bone need to remain adjustable. These growth plates, which are still flexible cartilage, enable it to elongate. Now for the section of our manual that becomes particularly complex. The femur has to grow in specific proportions. Remember, the muscle attachment sites are critical. Their placement, relative to each end, must remain in the proper ratio. Each growth plate needs to be informed independently so it can know when and how much to grow. Plus, at times, some of the superstructures will have to drift to a new position. All this is necessary to keep the bone growth in sync with the muscle growth. As a result, a girl can be just as agile at age 7 as a girl at 17. Sometime between the ages of 17 and 25, full skeletal maturity is reached, and there's no further growth in the length of the bone. But many more instructions remain. We need the bone cells to remain active for the rest of the owner's life. A fracture or break means new bone cells must be produced to make the repair. And if the bone is routinely supporting heavy loads, it will need to grow wider and denser for added support. Now, what we haven't covered yet is the interior, where we have the dense network of blood vessels and the spongy bone marrow, which produces the components of blood. Of course, the femur is just one of more than 200 bones in your body. The cells in each bone have their own set of instructions. Why? Because your bones develop in different ways and at different times. Finally, the skeletal system coordinates with our many other systems. We already saw the muscular system. Your body's circulatory system receives its red blood cells from the bone marrow, and your immune system uses the vital white blood cells, which are also produced in the bone marrow. Your nervous system is obviously critical to the transmission of information. It too is in constant conversation with the bones in your skeletal system. So, building even a seemingly simple bone is immensely more complicated than you might think. In fact, many consider the femur a marvel of engineering. The coordinated three-dimensional plans clearly show foresight, with an end goal of an optimized bone interconnected with an optimized system. Again, Each cell knows its role in the process of growing and shaping the femur to exact specifications, in specific locations, on a prearranged timetable, with updated instructions over its entire life. But the same can be said for your other organs, where every cell is purposefully arranged according to a well-laid plan. When you look at DNA, this remarkable molecule, able to store and retrieve information, an obvious question comes up. Where did it come from? For decades, scientists have discussed when and how the source code of life might have begun. In the distant past, did chemicals randomly organize over time into higher forms of complexity? After all, we see examples in nature where chemical reactions sometimes grow orderly and beautiful shapes. But in the world of biology, we know that working structures are possible only through the specific code that determines their form and function, not from randomly arranged chemicals. And as for the concept that DNA might have developed gradually in small steps, there's the obvious issue of the origin of proteins. We know that without the instructions from DNA, it's impossible to make proteins in a cell. At the same time, DNA instructions can't be accessed without proteins. That stops this chicken and egg speculation in its tracks. Instead, we know that an item that is well designed implies a skilled designer. So where did all this information come from? The evidence does not point to happenstance. Source code with the sophistication we've been talking about has never been known to arise without a programmer, an intelligent agent. That is, the designer and supplier of the information that is the foundation of all life. Perhaps more than we'd care to admit, we've become pretty dependent on automation. Robots do thousands of industrial tasks. What's fascinating is where automation is headed in the future. Developers are making headway towards swarm robots that will communicate and collaborate to solve problems, and patching robots that will autonomously identify damage and make repairs. It's exciting stuff. But in this episode, I'll be taking you into a world where this type of automation already exists. A place maintained by machines that are the envy of every scientist and engineer. You guessed it. I'm talking about the most technologically advanced place on planet Earth, the cell. In previous episodes, we looked at molecular machines that build and assemble the cells in your body. Today, we'll look at machines with a different task. Their job is to make repairs in your cells. Inside of you, there are trillions of microbots working hard to fix what's broken in your cells. Without them, your body would break down in a very short time. And unlike your air conditioner that doesn't get fixed until the repairman arrives, the molecular machines in your cell are making repairs 24/7. They do the work, and you can keep running without even knowing there was ever a problem. So let's move inside where these machines are found all over the cell. We'll be visiting the cell membrane, the cytoplasm, and the nucleus, which is our first stop, and the home to DNA. DNA is the carrier of the genetic information that helps make each of us who we are. One secret of the cell is that things go wrong with your DNA on a continual basis. In most cases, these problems have to be fixed. Here's what happens. As we saw in previous episodes, DNA consists of 4 different kinds of chemicals called nucleotides. Like letters in a sentence, their sequence spells out instructions for the cell. An error in the instructions can result in a defect. This is a critical issue when the cell replicates. The DNA has to be copied accurately so the sequences are identical. Copy errors can have serious consequences. Now we'll look at the remarkable mechanics of DNA duplication and repair. To help us follow the process, we'll be portraying the many machines in the cell as, well, machines. Before DNA can be copied over, it has to be unzipped by a machine called helicase. With the strands separated, we can begin the DNA duplication process. A truly remarkable machine called polymerase adds the complementary nucleotides to the growing new strand. The template is A with T and G with C. When all goes according to plan, the strand is completed with no distortion to the code, and you have a perfect copy. But now for the rest of the story. As I said before, things do go wrong with DNA. Actually, a lot of things. Now we'll go behind the scenes where myriad robots maintain DNA's integrity and keep you alive. Here's our first challenge. It turns out polymerase has a serious weakness. By itself, it isn't able to hold on to DNA very tightly. After adding just a few nucleotides, it falls off. You can't copy what you can't hold on to. So right off the bat, we have a major obstacle. But fear not, as we're going to see throughout this video, the cell is filled with gadgets that solve problems. In this case, we have this very cool clamp, perfectly designed to help polymerase keep its grip. But this donut gadget might seem odd to you. How does it fit around the DNA? Enter another amazing molecular machine called the clamp loader. It pulls apart the clamp and wraps it around DNA. Now the polymerase can merrily slide along the DNA, making a new copy. But now we have another problem. Sometimes an incorrect nucleotide gets added to the sequence. Fortunately for us, polymerase isn't some ordinary, undiscerning robot. It's a very fine proofreader. The polymerase will actually sense the error and immediately step back to cut off the incorrect nucleotide. It then proceeds as before, adding more nucleotides to the end. Leaving nothing to chance, some machines double-check for uncorrected mistakes. If mismatched nucleotides are found, a repair team cuts out the region. Polymerase fills it with fresh DNA, and another machine, ligase, stitches the patch into the DNA chain. At this point, I think it's important to point out that many comparisons have been made between machines made by humans and the machines found in humans. Clearly, we are an assembly of many sophisticated mechanical parts. So does that mean you and I are just machines? No, of course not. Machines are arrangements of parts that can be used for a task. They exist for the good of something else. But people and other living things exist for their own good, for their own purposes. The living organism is the being for whose good the machines of the cell work. Now, onto the cytoplasm, which has some of the most astonishing machines of all. The cytoplasm is busy with millions of protein machines, and sometimes they can get jostled and damaged. This transporter is carrying a lipid to the cell membrane. Usually it delivers the cargo smoothly, but today a collision occurs. The misshapen transporter now poses a hazard, and it must be taken out of service. And this proteasome does just that. Its job is to dispose of whatever is dragged into it. Destruction machines roaming freely in your cells? Not a comforting thought. But the proteasome has safety caps on both ends. Healthy machines are almost never consumed. So how does the proteasome know what to shred? The job of another set of micromachines is to recognize disabled machines and place a tag on it called ubiquitin. It's kind of like those signs you see on junked cars next to the road. The proteasome is drawn to the tag direct, but the opening is too narrow. No problem, the proteasome cap straightens it out, threading it inside where it's broken down into fragments by prods that break chemical bonds. These fragments are further reduced to amino acids, which the cell can reuse to build new machines. Interesting. Our cells have been doing recycling long before it became popular. Removing the trash is not a glamorous job, but it is a critical one. If rubbish builds up on the streets, all manner of health hazards can ensue. Similarly, a cell with a defective proteasome system is at risk for many diseases, including Parkinson's and other neurodegenerative diseases. And finally, the cell membrane. As with DNA in the nucleus and proteins in the cytoplasm, maintaining the integrity of the cell membrane is a constant struggle. At times, tears occur, causing internal contents to leak out and external material to flow in, leading to cell death if not quickly stopped. And what's sort of disturbing is that these tears don't just happen occasionally. At any one time, up to 10% of active muscle cells can have tears or holes in their membranes. As many as 12% of nerve cells. You don't want to lose those. And as many as 25% of muscle cells in the heart have disrupted membranes. That's a lot to repair. Yet despite such challenges, cells can live for months, years, even decades due to a range of membrane repair mechanisms. Here's just one of them. Your cells recognize a hole when an abnormal amount of calcium flows in. Sensors detect the increase, triggering an alarm that signals the repair crew to act. These remarkable hollow spheres made of membrane material come to the rescue. When they land, the material flows together and tension is reduced. A perfect patch is created without you ever knowing you had a problem. Cells with holes in them can't survive longer than a few minutes without being repaired. The cell's fate is similar without DNA repair bots or proteasomes doing cleanup. So as we wrap up this episode, we need to address a glaring question. When early single-celled life inhabited Earth, how could it survive until repair machines evolved to maintain it? To answer that, we need to first remember that repair machines are separate, independent systems from the machines they repair. Tools needed to repair a car are not parts of a car. Tools needed to repair a photocopier are not parts of a photocopier. The upkeep of complex machines requires forethought, planning, and customization. In the very same way, since the beginning of life, cells required upkeep by machines made precisely for their needed repairs. So it's the old chicken and egg problem. You can't have one without the other. The first life necessarily had a complete maintenance system. And just like so much else we've seen about the cell, its remarkable repair machines, which sustain you every moment of your life, are products of forethought and purposeful planning. That is, they are products of intelligent design. I'm Mike Behe, and I look forward to seeing you on the next episode of Secrets of the Cell.